I'm Charles Mullighan from St Jude Children's Research Hospital, and I'm joined by my panellists Kathryn Roberts from St Jude and Anjali Advani from Cleveland Clinic. We're at the first international workshop for acute leukemia, and we've just had an excellent session on recent insights from the genomic analysis of acute lymphoblastic leukemia. We've discussed insights into pH-like acute lymphoblastic leukemia, into early T-cell precursor leukemia, and then a broader discussion of recent insights into the landscape of ALL. I'll start by turning to my panellist, Kathryn Roberts. Could you give us a summary of recent insights of pH-like ALL from your talk?
Sure. So we've identified a new subtype of pH-like ALL. These patients have a gene expression profile similar to the more well-known BCR-ABL positive ALL cases, but they lack the BCR-ABL fusion, and so suggesting that they have additional kinase activating alterations in these patients. And this is very important clinically because we think if we can identify the genetic alterations that are present in these patients, we may be able to target those with tyrosine kinase inhibitors, as has been shown for BCR-ABL. And importantly, as I should mention, these patients have a very poor outcome compared to non-pH-like ALL. So the addition of tyrosine kinase inhibitors, or TKIs, may improve the therapeutic outcome for these patients. One important observation that we've made is that the frequency of this subtype actually increases with age, to account for approximately 25 to 30% of adults with ALL, in a similar pattern to BCR-ABL positive ALL. So that's an important observation, and obviously these patients are a very important clinical entity. We've done several in vivo studies to show that these patients may respond to several classes of tyrosine kinase inhibitors, and we've also done very detailed genomic landscape studies to identify the different genomic alterations. And we've identified two main subsets. One group that harbours what we call ABL class alterations. These include fusions of ABL1 itself and a couple of other different genes that are responsive to ABL inhibitors, such as imatinib and dasatinib. And we think that adding these agents to a chemotherapy backbone, similar as what has been done for BCR-ABL ALL, may be able to improve the outcome and kind of synergize with the chemotherapy to improve their event-free and overall survival. And then we have a second group of patients that harbour alterations that activate what we call JAK-STAT signalling. And so again, we've identified many different alterations that contribute or converge on these signalling pathways, rearrangements of a gene called CRLF2, JAK2 itself, the erythropoietin receptor, which is hijacked by the immunoglobulin locus to be aberrantly expressed in B-cell ALL, and a couple of other different genes in the JAK family. And we think at least the first set of agents that we will try to improve the outcome for these patients are JAK inhibitors themselves that target this pathway. And so I think the main take-home messages are, despite the complexity at the genomic level of these different alterations in this subset that we call pH-like ALL, we have a limited number of agents that can be deployed or implemented to treat these patients. And these trials are ongoing, so within the next few years we will be able to assess the broad clinical activity of these agents. We do have anecdotal reports of patients being treated with these agents showing a great response, particularly with the ABL class alterations. So we're very hopeful that this will be a big improvement for those patients, and we're still waiting on data to come through for JAK inhibition. We also discussed additional therapeutic combinations beyond TKIs, and looking, for example, at newer generation TKIs, also combining different classes of tyrosine kinase inhibitors, those that target JAK-STAT pathway and also the PI3 kinase pathway. And we also looked at agents that target completely different pathways such as the apoptotic pathway, so inhibitors that inhibit the BCL2 pathway, for example, and induce apoptosis. Can we combine those kind of agents that are apoptosis inducing with the TKIs and see an improved response? And another class that's very important, it's being actively pursued in ALL in particular and has shown great promise alone, immunotherapies such as monoclonal antibodies that target proteins that are expressed on the cell surface of B-ALL cells. Can we maybe, is the outcome improved with these newer agents, and potentially can we layer TKIs on top of those as well? So I think that's kind of the take-home messages. There are still some entities, for example, CRLF2 rearranged, that are present in adults at a very high frequency. It's probably the most important group that we need to target, but I think we're definitely making great improvements. Now that we know the genomic landscape, we have an arsenal of different agents that we can employ for these patients.
Okay. Anjali, what are the key points of your presentation on ETP ALL?
So with ETP ALL, typically it's been considered to have a poor prognosis, and as compared to the other TALLs. And so there's been a lot of question about how these patients should be treated, how we can improve outcome. And there's actually been a lot of nice research really looking at the biology of this ETP ALL, showing that some of the biology actually overlaps with the myeloid subtypes such as AML, and that there are specific mutations such as DNMT3A and FLT3 that are uniquely expressed in this subtype of TALL. And so in the future, those may be targets for therapy. So I think kind of getting back to what Kathryn was talking about, it's a little bit harder because this is a very small subset of patients, but really trying to better understand the biology and potentially trying to target some of these other mutations may be important. It's not so clear as with PH-like ALL that these mutations are the only ones that are important. So I think that's the harder part of this. In the ETP ALL, a smaller proportion will also express Notch mutations, so treatments such as the Notch inhibitors may also be important. And so a lot of what we focused on today was really in the early studies that have been done showing that the prognosis of ETP ALL was worse than other patients with TALL. The question was, should these patients be transplanted up front? And there's been some nice work been done by the Children's Oncology Group in a clinical trial they performed in TALL, where they performed risk assessment. This was the AALL0434 study, and they used risk assessment to basically, based on MRD and bone marrow response, stratify patients. And within that study, then there was also a randomization to high-dose methotrexate or nelarabine. The patients that were very high risk, almost all those patients received high-dose methotrexate and nelarabine. And then in that study, they looked at all TALL patients, but in there, because they had such a large subset of patients, they specifically looked at the ETP subset and found actually that their prognosis was no different than the other patients with TALL. And so it suggests that using a treatment strategy tailored based on MRD and risk may be able to abrogate the negative effect of the ETP subset. There also has been a nice study, a transplant study that's been published by Bond in the Journal of Clinical Oncology. They took a little bit of a different approach. This was an adult study through the French group, and basically in that study they transplanted only those patients that had high-risk features such as MRD or were initially resistant to therapy or CNS disease. And by selecting those patients that were high risk and transplanting them, again, they were able to show that their outcomes were similar to those with TALL. And so I think, the major things to consider are really, it looks like stratifying therapy really based on minimal residual disease is probably important. Second is that there may be new agents such as nelarabine, and potentially incorporating those into upfront therapy may improve outcome, may potentially decrease the number of patients we need to take to transplant. I think that's going to be a question that's really going to need to be answered in further randomized studies, which hasn't been done yet, and except in the Children's Oncology Group study. And then the third question is really going to be whether we can potentially target some of these other lesions such as DNMT3A and FLT3, much as we've done in AML, and whether that may potentially improve outcome.